Hello, thanks for joining this poster session. My name is Raquel Dias. I'm an assistant professor at the Microbiology and Cell Science Department. Today, I'm going to talk a little bit about two of the main projects that I have in my lab, where we apply artificial intelligence in the field of clinical genomics for answering different and complex questions in those fields. So as I said, I'm going to talk a little bit about two of the main projects that we have in the lab. The first one is called a rapid reference-free human genome imputation with denoising autoencoders. It sounds a little bit complicated, but I'll do my best to try to simplify it and explain it. The second one is an explainable AI method that integrates gut microbiome data, host genomics, and environmental factors for the early detection of type 1 diabetes. So in the field of clinical genomics, there are usually two types of common data acquisition techniques. The first of them is the whole genome sequencing, where you can obtain the complete set of genotypes in the human genome and identify single variation in the genome. The second method is genotype array that only identifies a few regions of the genome. Of course, the whole genome sequencing is the best method, but it's very expensive. It's about 10 times more expensive than the genotype array, and it also takes longer to generate the data. So most of the clinical studies, all those large cohorts comparing cases and controls and try to predict the genetic risk for complex diseases, they use genotype array data, and they apply imputation, which is a method that based on a reference set of whole genome sequencing haplotypes, they try to use a method to guess what are those missing values in the study genotypes. Here, for example, we have the genotypes, the ATCGs of our genome represented as zeros, ones, and twos. The language that is supported by the machine learning techniques. So the typical imputation methodology available today is based on hidden Markov models or HMMs. And these methods depend on very large reference panels that have a lot of access restrictions, which makes these methods many times inaccessible to the end user. So what we are proposing here is to replace those statistical methods, those hidden Markov model methods, by a more AI-based approach. And in this approach, we start with the full set of genotypes. We mask the data gradually in each training iteration of our models. And then we utilize autoencoders, more specifically denoising autoencoders, that will learn the most representative features in this data. And at the same time, it will learn to unmask those masked genotypes and restore the original information in the data. Then we compare the imputed genotypes generated by our autoencoders to the original ground truth data, the whole genome sequencing data that we had. Once the model is trained, then it can be used to make new imputation on genotype array data by the end user without needing a reference panel. So we developed and trained our autoencoder approach across chromosome 22 of the human genome. And we compared those results to the top 3 most cited imputation tools available, BIGO, Minimac, and Impute. And through these results, we can see that the autoencoder seems to outperform all the other hidden Markov model-based methods across several minor allele frequencies in the population. So even when we have very imbalanced genotypes in the population, we still see some advantage of using the AI method versus the traditional HMM-based approaches. And these results are pretty consistent across different data sets. For example, here, the MESA data set that is very multi-ethnic, and the Welderly data set that is more European ancestry predominant. And also, we see the results are consistency across different genotype arrays as well. So for example, here we are testing the Affymetrix array, the UK Biobank Axiom array, and the Omni 1.5 million array. And all of them are showing that the autoencoder consistently outperforms the typical HMM or hidden Markov model methods. The second AI system that I wanted to talk about today is more focused on using multi-layer, multi-omic types of data and integrate that data for then predicting complex disease outcome. In this case, we are focusing on type 1 diabetes for our main project, which is an autoimmune disease that affects millions of babies every year around the world. And it has many possible complications, including kidney disease, infections, glaucoma, shortened lifespan, early premature death in babies diagnosed with type 1 diabetes. So it's a disease of critical interest. And any approaches that can improve the early detection or mitigation of this disease are very welcome. And also the scientific literature shows many evidences indicating that there is some association between the gut microbiome or the microbiota of the patients and the autoimmune diseases. So potentially the gut microbiome could be used as a source of diagnosis. So our main project starts with trying to detect associations between the gut microbiome and autoimmune diseases. And later we're going to try to integrate genetics, metabolites, medications, and all sorts of environmental factors with the microbiome data to try to boost the power of our model when trying to predict type 1 diabetes, and probably expanding these models for predicting other autoimmune diseases. And by using AI interpretability techniques, we're not just going to get accurate predictions from these models, but we're going to understand exactly why these models are doing those predictions, so we can be more helpful and more informative to the domain experts and clinical practitioners. And the data set I'm going to be using here is the AVIS data set, all babies in Southwest Sweden. It's about 1,600 subjects. And here for these preliminary results, we're going to be merging type 1 diabetes and celiac disease as a group of autoimmune diseases for boosting the power of the model, for increasing the total number of cases versus controls. So by using our explainable AI technique, we can see that when we model the disease without using microbial markers or the microbiome as predictors, we only use genetics and environment, we can achieve mean accuracy of around 60% after doing 5-fold cross-validation. But when we add microbial markers as predictors in our model, we observe a boost in accuracy. The model that was 60% accurate is now has now a 73% of accuracy. So this indeed confirms the contributions of the microbiome when predicting autoimmune diseases. And once we validate this model in an external data set and we expand it to a larger training data set, this model shows some potential of becoming a tool with some clinical utility for early prediction of these diseases. And finally, when we use the AI interpretability techniques in our most accurate model, we can see that many of those microbial biomarkers perform interactions with other microbial and non-microbial markers that have a significant impact on the model output, on the prediction of the risk for the autoimmune disease. Another interesting thing is that, for example, Gordonibacter, it has many risk increaser effects, but when it's combined with other biomarkers like the genus Sarcina here, it becomes a protective biomarker. So those were the two AI systems that I wanted to explain a little bit about today. As a conclusion, we can see that both systems can accurately perform genomic imputation and disease prediction. Of course, there are multiple places where we can improve the system and further expand it. For example, for the imputation system, we are planning to expand the system to non-human species. So far, we only worked on imputation of human genomes. There is a lot of potential to expand this to other non-human species. And the disease prediction system, there is a lot of room for future integration. For example, we can integrate with the imputed data from the system 1 to boost the accuracy of our models. We can also include metabolomics that we haven't included yet, and proteomics as well. So we can find interactions between the host metabolism and the microbiome, for example, and how these interactions affect the risk for autoimmune disease and for type 1 diabetes. And we can definitely expand the system using other data sets that have much larger sample size and much larger ratio of type 1 diabetes cases. For example, the environmental determinants of diabetes in the young, the TEDdy study that has a sample size of 8,000, and all the individuals have high risk for type 1 diabetes. So it will be very interesting to adapt that workflow to this larger data set. So that's all that I wanted to share today. Thank you very much for your attention, and I will be happy to meet you in the poster session to answer any of your questions. Thank you very much.